Okay, so welcome back from your break. Let's take a look at the concept check. Why are bone marrow cells considered a good target for human gene therapy? The answer is really easy, and it has something to do with what I mentioned about what the bone marrow cells do. They're constantly growing and dividing for the whole of your life, which means that any genetic change that you make to these, any genetic correction that you make to these, will last for the length of your life, so long as it takes. So if you're using the viral vector, one of the things that is really important is that the virus infects the cells and that the correction sticks. Because once you've infected the cells with that virus, you can never reinfect them again. It's a one and done. And that's part of the problem with using the viral vectors at the moment. If the fix does not take, then you automatically rule out a second chance, because the virus has already infected the cells once. You can't reinfect a second time. The cells will not permit that. So the reason for targeting bone marrow cells is simply because they are constantly growing and constantly dividing and providing a source of cells that move out into the body for the whole of your life. Remember, they're producing your red blood cells, your white blood cells, and the other cellular components of your bloodstream. And if you recall from last week's chapter, some of those cells can also develop into cells in the heart muscle. They've been used to repair damaged hearts in rats that have had induced heart attacks. And they can also develop into nerve cells. So they have the possibility, because of their nature, to develop into components of other parts of the body as well. So if you could take those cells that have been genetically modified using the gene therapy technology, you may actually be able to introduce the repair into other tissues as a consequence of what those bone marrow cells can actually turn into, or cells derived from the bone marrow cells can actually turn into. So they're a really good target for human gene therapy. So the next thing that we want to do is look at the use of DNA in forensic science. I believe that's what the heading here is. Yeah. So it's forensic science utilization. So the most common thing that we know, that, or you've probably known, that DNA is used for is DNA profiling, a DNA fingerprint of an individual. So what do we mean by that? What is a DNA fingerprint of an individual? Well, this is how we utilize the DNA profile. You take the DNA that you've been able to isolate from the biological material left at the crime scene, and there's always going to be biological material left at the crime scene, unless the person carrying out the crime wore a complete head-to-toe hazmat suit that covered everything and prevented any biological material of any sort from escaping while they committed the crime, and they'd probably be a little bit obvious if they did that. There's always going to be biological material left behind. And as I mentioned at the beginning of this chapter, we need now remarkably small amounts of biological material in order to be able to do a DNA profile. And the reason why is because of the second step, DNA amplifying. And we'll talk about that. It's a process called polymerase chain reaction. And it has changed the nature of forensic profiling using DNA completely. I mean, it's changed so much about our use of DNA and DNA technology, but in the realm of forensic science, it's really changed everything. And the reason why is because it gives us the ability to make huge copies of selected regions of the DNA from just a small amount of DNA. Don't need very much. As I said, 15 cells worth. That's a really, really, really small amount of DNA. You may be familiar with what a milligram is. It's a very small amount of mass. Well, here, 15 cells worth of DNA is less than 1 microgram. It's about 1/10 of a microgram. So a microgram is 1/1000th of a milligram. And a milligram is 1/1000th of a gram, and a gram is 1/1000th of a kilogram. So we're talking about a really, 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 really small amount of DNA. But using the PCR technology, we can amplify that. We don't need to amplify all of the DNA, we just need to amplify selected regions of it. So we amplify the selected regions of the DNA that we want to look at. And we then, if we have some suspects and we have the legal authority to get a DNA sample from them, which according to the Supreme Court, we do, or at least law enforcement does, then you can get the DNA sample from your suspects, and you can do the same thing, amplify the selected regions of the DNA that you want to amplify. And then you carry out a process called gel electrophoresis, which we'll look at in a little bit more detail. We're going to look at all of these in a little bit of detail. And then you compare the DNA from the crime scene against the DNA of the suspects. And because there is such a very, very low probability of two people sharing exactly the same DNA profile and not being at least blood related. So it's possible that if your blood relative, brother or sister primarily, but a family member left biological material at a crime scene, and you are suspected of being the perpetrator, your DNA may be sufficiently close enough in a match because you are a blood relative of the person who actually did it, that you may be accused of being the person who did it. In fact, that's one way in which some perpetrators of old crimes have been caught, not because they have a sample of the DNA of the individual who is suspected of doing the crime, but because they have access to DNA sequences from relatives of the individuals that they suspect did the crime. More of that a little bit later, because that's a little bit interesting there too. So by comparing the DNA sequences that you generate from the crime scene DNA and the individuals that you think are your suspects, you should be able to identify the individual who left behind the biological material. Now, there is another issue with this, and it's a legal issue, and there is a group called the Innocence Project, which we talk about a little bit later in this section, that has been using DNA technology to demonstrate the innocence of people who have been wrongly charged with crimes, including people who have been on death row for a number of years. One of the issues with all of this is that if you do this gel electrophoresis and you find that the DNA of an individual, one of your suspects, was at the crime scene, all you can really say is that that individual's biological material was at the crime scene. You can't say definitively that the individual was at the crime scene. I know this sounds like splitting hairs legally, but it is an argument that is very legitimate. And the Innocence Project has actually used that argument successfully in court to gain the release of people who, as it would turn out, were actually innocent of the crime, that other people had actually carried out the crime. with further investigation on the behalf of the police force. So this is not a perfect system, but it's a really, really good system nonetheless for identifying individuals who may have committed crimes. So let's talk about the polymerase chain reaction for a moment. With the polymerase chain reaction, it's basically taking advantage of bacteria's ability to replicate DNA very, very quickly. And the fact that some bacteria, because of their ability to live at high temperature, can also replicate their DNA at high temperature. Now, the original PCR system involved taking the DNA, placing it in special little cap test tubes that were designed for this purpose and placing them in PCR machines. Now, originally it was all done in water baths and things like that when the original technique was developed by a scientist by the name of Kerry Mullins. And I believe he's just received a Nobel Prize for the development of the PCR system, or was included in a Nobel Prize award. There's some major award that was given to Kerry Mullins just recently. I remember seeing a news piece on this. Anyway, there are now machines that'll do this. And so you put in the original technique, you started at room temperature. You heated the DNA to around 90 degrees Celsius. Now, why you do that is because the hydrogen bonds that hold the two polynucleotide strands of the DNA together actually break at 90 degrees Celsius. And so the two strands break apart. You then have included in your mixture a piece of DNA that is complementary to a small section of the piece of DNA that you're trying to amplify, and that acts as a primer to allow DNA polymerase then to go ahead and copy the two single strands that you've now created by heating the DNA and causing them to fall apart. And in the original technique, this was done after the DNA cooled back down again. And as it cooled down, that allowed the primer to bind and stop the two strands from sticking back together again and allowed DNA polymerase to do its thing at a lower temperature. Then you would reheat it, the strands would fall apart, the primers would then reattach, DNA polymerase would do its thing. And this would run for 24 hours to amplify the DNA. And you needed a reasonable amount of DNA in order to be able to do this. Today, as I've indicated on a couple of occasions, you need a lot less DNA, and you don't need to worry about the heating and cooling, heating to high temperature and then cooling back down to room temperature again. Because we know of bacteria that live at temperatures of 80 to 90 degrees Celsius. And so because they live at that temperature, their DNA replication machinery must also function successfully at that temperature. So today, rather than having to heat and cool to high temperature, back down to room temperature, back up to high temperature, back down to cool temperature, everything can be done at high temperature. And the difference in temperature is increased to get the DNA to fall apart versus cooling down to allow everything to stick back together again and the polymerase to work is only about 10 degrees. We use bacteria that thrive at around 75 to 80 degrees in the hot water pools in Yellowstone and other places where bacteria are known to live in hot water pools. And so we use the DNA replication machinery from those bacteria. And because you don't have to heat and cool so much, the temperature difference between hot and cool is only a small amount. You don't have to wait anywhere near as long for it to cool down to do the copying. And so the cycles occur much more frequently. And now instead of taking 24 hours, you can amplify the DNA in 3 or 4 hours. From a much smaller amount of DNA, you can do the amplification much faster because you're using a high temperature based system. So this is the reason why people are really interested now in going back and looking at stored biological material from older cases and using PCR technology to amplify the DNA from those materials, and then take advantage of other DNA technology that we're going to talk about as we continue on here. So once you've got this amplified DNA, then you can do all sorts of things. So you can look for the presence in, for instance, of HIV. One of the very sensitive techniques for looking for the presence of HIV in blood is based on PCR. The amount of HIV that is detectable in the bloodstream by antibody techniques means that there needs to be a fairly substantial amount of HIV in the bloodstream, because antibodies are not terribly sensitive. But PCR DNA technology is at least 150 times more sensitive than the antibody system. So it allows you to detect lower concentrations of virus in the bloodstream, which is always really important. And in fact, in our current situation with the COVID virus, there are in fact two different types of COVID virus tests. The quick and easy one is built on antibodies. It's what's known as an ELISA test, E-L-I-S-A, ELISA. And it uses antibodies to detect the presence of the virus, antibodies against the virus to capture the virus, and using a color reagent to indicate whether the virus is present or not. So if you see color, you know the virus is present. If you don't see color, then the virus perhaps is not present, but it's not terribly sensitive. The more sensitive test for COVID is the PCR test. And they actually have referred to it in the news as the PCR test for COVID. But that takes 24 to 48 hours. So if you want a quick and dirty instant result, you use something like an ELISA test, an antibody test. But if you want a more accurate and more sensitive test, then you use a PCR test. So it has relevance to today. For those of you who are interested in doing a blood donation, I can tell you, as part of the extra credit, your escrow account, if you want to boost your escrow account to get a better extra credit value at the end of the semester, which is only a week away, then as part of your blood donation, the blood bank runs a series of antibody tests checking for the presence of different contaminants like HIV. And since we've been in lockdown, so for about the last 12 or 13 months or so, the blood bank has been running the COVID virus antibody test against blood tests. And this is one of the reasons why I am certain now that my vaccination, because I got the Pfizer vaccine some months ago, I'm certain that my vaccination actually took, because when I gave my blood donations, because I am a regular blood donor, my antibody tests came back negative. And so I had 3 negative tests. And then the very first test that I had after, or blood donation that I gave after completing my vaccination protocol, and knew that I was fully vaccinated 2 weeks out after the second shot, my antibody test came back positive. So it was negative, 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 then after the vaccine, positive, telling me that I'm producing antibodies to the COVID virus. And to the best of my knowledge, I've not had COVID unless I had the asymptomless version. So my production of antibodies is probably as a consequence of my vaccination. But again, that's using the ELISA test, not the PCR test. The PCR test is much more sensitive because you can detect lower concentrations of the virus because you're amplifying the genetic material of the virus to higher levels to allow you to determine whether it was present or not. So then, how do we go about comparing DNA samples once we've amplified them? Well, it turns out that within our genome, the vast majority of the DNA is non-coding. Only about 3% of our chromosomes actually contain genes that are functional and are utilized by humans. The other 97% has, until recently, been commonly referred to as junk DNA. But we now recognize that it is probably really important. Now, some of it, and this is going to sound a little bit scary, but you don't have to panic here, some of it is actually viral DNA. The good news is that viral DNA has been incapacitated. So these are non-functional viral DNA pieces. We know that they're from viruses because we recognize the genetic signatures, but the genes have been disabled. They're no longer functional genes. We call them pseudogenes. They were once functional genes, but they have been inactivated. But we also know that within this non-coding region are many sections of DNA that are involved in regulating gene expression. And we talked about some of that in the previous presentation. And within that region of non-coding DNA, there are what are referred to as short tandem repeats. And repeats in DNA are really common. We know that there are lots of repeated sequences in human DNA. At the ends of chromosomes, there are repeated sequences. These are called telomeres. They are there to be sacrificed every time the chromosome is copied, because one end of the chromosome, the lagging strand end of the chromosome, is going to get shorter every time the chromosome is copied because of the way it happens. And so the telomeres are there as repeated sequences. Each telomere is 6 nucleotides long, and you could have up to 175 repeats on the ends of your chromosomes. They're there to be sacrificed. These short tandem repeats, however, occur within the body of the chromosome. And these are not only repeated sequences, they're also inherited sequences. And your particular set of short tandem repeats are a product of the short tandem repeats that you inherited from your mother and your father. So they have a familial origin. This is the reason why we can identify family members using DNA profiles. This is the reason why when, for instance, the remains of a military individual or the remains of individuals that were killed in the collapse of the World Trade Center back in 2001. This is the reason why they asked relatives to provide DNA sequences, because they could use the DNA profiles, the short tandem repeat profiles of the relatives to identify the remains of the individuals on the basis of the fact that these have a genetic component. They have a familial component. So they may not be identical, but you can tell that they are the product of family connections through inheritance. So it is the short tandem repeats and the combination of short tandem repeats that each of us have that are, in essence, like a fingerprint. They're unique to each individual. Yes, they share a familial similarity, but they're not a familial identity. They're not identical to each other. They are truly like a fingerprint. They're unique to each individual. So your particular combination of short tandem repeats are going to be different from your brothers and sisters. Can we tell that you are brothers and sisters on the basis of those STRs? Yes. We can tell also who your parents are on the basis of those STRs. But each of you are going to have a slightly different combination of those STRs. And that's how we identify them. So here is an example that gives you some idea of what you might be looking for. So here is a sequence, and the tandem repeat simply means that you have a repeat of this sequence of nucleotides back to back to back in a chain, and you have a certain number of these repeats as a consequence of what you've inherited from your parents. At a different location, you have another sequence of nucleotides that is repeated multiple times, another set of tandem repeats. But one individual, the suspect, has a different number of repeats than the number of repeats at this location that were found in the crime scene DNA. So as much as this person might be a suspect, this difference, this sequence is the same, but this sequence is different. This individual did not leave this DNA behind. These are DNA from two different individuals. That's how we do the fingerprinting. We look at a number of different locations. So here we're just looking at two different STR sites, but we look at multiple STR sites. And it's because you have multiple sites with multiple repeats and different number of repeats depending on what you've inherited from your parents and the combinations of the repeats that create the individual profiles. So here's the sequence of events that you would use to separate out the DNA fingerprints using this technique called gel electrophoresis. Now, the gels themselves are made out of usually a material like agar or another substance called polyacrylamide. It creates a gel-like material, and in that gel-like material are little holes that are filled with a solution that contains a lot of ions, positive and negative ions. When you apply an electrical charge across the gel, those ions migrate down the gel and carry the electrical charge through the gel. And if there are molecules that you apply to the gel that also have charge, those will migrate as well. And DNA happens to have charges on it. So you can cause the DNA fragments to migrate. But if the DNA is cut up, using restriction enzymes, for instance, which is the most common way to cut it up, into fragments, those fragments are not all going to be the same length. They're going to be different size fragments. And the fragments will move through the gel according to their size. The smaller fragments will move faster, the larger fragments will have a tougher time getting through the gel and so will move slower. So you take your DNA that you've amplified, you cut it up using the restriction fragments. And what you're looking for now is the presence of fragments that contain STRs. And the length of those fragments are going to be a function of the length of the STRs. And so it's really important to know which restriction enzymes to use to cut the DNA to get the fragments in the STR regions. And because of the length of the STRs, the number of repeats that you've got, the fragments are going to be of different lengths, and that's going to create your DNA profile. So you take that DNA and you apply it to the surface of the gel in one of these little wells. And you form the wells when you form the gel. You apply the voltage and the fragments of DNA start moving through the gel. And you have ways of either highlighting the location of the fragments. You can actually apply dyes that let you see the movement of the fragments, or you use markers that you add in that have fragments of different lengths, and you can see how far everything is moving on the basis of the marker fragments that you've got in. But at the end, you're going to highlight all of the DNA fragments in the gel. And there are different ways of doing that so that you can see where all the fragments are. And remember, each fragment is a product of the STRs that are present in it and the length of those STRs. And that creates a unique profile or fingerprint for each individual based on fragments and the STRs and the length of those STRs. And so now you can compare the crime scene DNA with your suspect's DNA and take a look and see if the fragment profile is the same or different. So here's an actual gel showing the various fragments. These are the standards that were used in the gel to see how far fragments of different sizes had moved. So the small fragments are down here, the big fragments stay up near the top. And then you've got the DNA solutions that were added in here. And then the forensic scientist is going to compare across these different lanes containing the different samples of DNA fragments to find the DNA profiles that match the DNA from the crime scene based on the banding pattern that you see in the gel. This is actually in a special gel carrier that can seal down, and you apply a vacuum to it that allows you to dry the gel, and the gel dries into what looks like a sheet of plastic, because that's what, I mean, polyacrylamide is an actual plastic. So when you suck all the water out of the acrylamide gel, it turns into a thin plastic sheet, which allows you to preserve all of these DNA fragments. And so when they introduce DNA evidence into a court, it's these dried gels and the patterns that are on these dried gels that can be introduced directly, or photographs of these. They may actually keep the gels in the research laboratories, in the forensic laboratories that generated these, but they'll present photographs of these dried gels as the DNA evidence. So again, we're just looking at another diagram that illustrates how to do this. So here's the crime scene DNA, the suspect's DNA, run it through the gel. Here are the fragments from the crime scene DNA. Here are the fragments from the suspect's DNA. Clearly, the suspect was not the person who left this biological material behind at the crime scene. So it is unlikely that this individual was at the crime scene. Otherwise, we would see a DNA profile identical to the suspect. So as I said, the Innocence Project has been working with DNA technology, in some cases using DNA technology to demonstrate the innocence of an individual, and in other cases arguing against the DNA technology that has cleared someone and indicating that in fact the DNA technology, because of the limits of the DNA technology that I was talking about earlier, did not actually rule an alternative suspect out as the actual perpetrator of the crime. The person who established the Innocence Project was actually one of the lawyers that defended O.J. Simpson during his trial back in the 1980s, and lawyer by the name of Barry Schenck. He got very interested in the use of DNA technology as a result of his involvement in the O.J. Simpson case, and decided to investigate further the use of DNA technology. And as a consequence of that, and of his investigations, he established the Innocence Project to use DNA technology not only to get people out of prison, but also in some cases to establish who was actually the person who committed the crime. So here's an example of an individual whom the Innocence Project worked with to get out of jail, but also use of the technology to establish who actually committed the crime. So this is Earl Washington. He was charged with rape as a consequence of witness statements. It turns out that at the time, the DNA technology was not sufficiently advanced enough to provide evidence for guilt or innocence. But by the time the Innocence Project started working with Mr. Washington, the technology existed to do a DNA profile with the limited amount of material that was available for generating a DNA profile. And this is the STR profile of the genetic material left behind on the rape victim. This is Mr. Washington's profile. And this is the profile of a second individual that the Innocence Project were pretty certain and that Earl Washington had actually suggested might have been the person who committed the crime. This is the profile of the second individual. Who do you think of as guilty of the crime? It's pretty obvious that Mr. Washington was not the person who committed the crime. In fact, Kenneth Tinsley was the individual who committed the crime because his STR profile exactly matches the DNA profile of the genetic material left at the scene of the rape. Mr. Washington's STR profile does not match in any way, shape, or form the DNA profile of the biological material left at the scene of the crime. There's no way that he should have been charged with the crime. And so as a consequence of the Innocence Project's work, Mr. Washington was released from jail. Mr. Tinsley wound up being charged with the crime. So this just gives you an indication of the statistics for the STR analysis. There are 13 specific locations within the DNA that are amplified to look at the STR sequences. And if you look at the chance of two individuals having exactly the same profile, chance is 1 in 10 billion to 1 in several trillion, which means if your profile matches the profile at the crime scene, at least that's your genetic material at the crime scene, so the chances are really good you were there. Perhaps you are the guilty party. This is the reason why police forces and forensic scientists really value DNA material. The ability to be able to amplify really small amounts, what in the past would have been impossibly small amounts to work with. That 15 cells worth, by the way, that's less than the number of cells that are attached to the base of a hair when you pull it completely out of the skin or completely out of your head. If you take a hair, if you pull it out correctly, you actually get the little gland that secretes the hair that comes out with it, and you have a little ball of cells at the bottom. There are more than 15 cells attached to the base of the hair. So if there's a single hair left behind with the follicle intact, that's the name of the little ball of cells on the end that secretes the hair, then there's enough genetic material in that follicle using PCR to amplify it, to generate a DNA profile. So you can imagine why forensic scientists and police forces and legal authorities everywhere value any small amount of biological material. And as it's pointed out here, if the DNA material is incorrectly handled, if it's mislabeled, if there are errors in the handling and the generation of the fingerprint, then it's possible that a misidentification, a misanalysis can occur. This is why the Innocence Project continues to work with incarcerated individuals, because there is always the possibility of error in the results. And if you can show how the errors have changed the analysis or influenced the analysis, you can either get a case reopened or you can get a person exonerated, get them released from jail. And that's particularly obviously helpful for people who have been incorrectly charged with a crime. So I mentioned the use of DNA profiling for the World Trade Center. Another interesting use of DNA profiling is the case of Thomas Jefferson and Sally Hemings. For many, many years, there were a whole line of African-American individuals who claimed descent, direct familial descent, from Thomas Jefferson via one of his female slaves named Sally Hemings. It was known that Jefferson did have a relationship with Sally Hemings, who was herself the daughter of a good friend of Jefferson and one of that man's female slaves. So Sally Hemings herself was the product of a slave owner having a relationship with one of his female slaves. And these African-American individuals claim that Jefferson had fathered a child with Sally Hemings and that this line of individuals was descended from that child of Jefferson's via Sally Hemings. The white descendants of Thomas Jefferson denied this and argued vociferously, vocally, very vocally, that this was impossible, that Jefferson, number one, didn't have a relationship with Sally Hemings, and even if he did, there was no offspring as a consequence of this, and therefore there was no African-American lineage descended from Thomas Jefferson. Eventually, several individuals, both from the African-American lineage and the white lineage, had DNA profiles done. And it turned out that indeed, the African-American line was descended from Thomas Jefferson. He really did have a relationship with Sally Hemings. He really did produce children with her, and that at least one of the children, we're not entirely sure how many children they had together, but at least one of the children was the ancestor of the African-American line of individuals that claimed descent from Thomas Jefferson. In the face of that DNA evidence, no longer could the two lines of descendants from Thomas Jefferson argue that they did not share a common heritage. And now they do acknowledge a shared common heritage, and they are now act as the common descendants of Thomas Jefferson. But it took DNA technology to demonstrate that. And periodically, individuals have appeared at various times claiming descent from famous European royalty. So Marie Antoinette is one example. There have been a number of individuals who have claimed to be descendants of the Russian royal family that was executed by the communists in 1918 when they took control of Russia. But DNA technology over time has demonstrated that in fact no one has been able to demonstrate through DNA descent from either Marie Antoinette or from the Russian royal family, and it is considered that there are no existing survivors of either of those individuals. Another way that DNA technology is now helping is to identify animal-derived material that has been taken from animals that are protected. So an example of this is ivory from African elephants. There are some forms of ivory that are still legal to take, but not elephant ivory. Ivory is in fact just tooth material. And so the tusk of an elephant is actually an overgrown tooth. And you can get ivory from other sources, walrus tusks, other tusk-like material constitutes ivory. And there is DNA in that ivory, just as there is DNA attached to a human tooth, and a human teeth can be used to provide DNA for profiling. So ivory can be used as a source of DNA for profiling. You can get DNA out of preserved skins. DNA has been used to demonstrate that the whales that have been taken by the Japanese whaling fleet that is still allowed to take a limited number of whales have at times been taken from protected species of whales or protected populations of whales. This is the reason why the International Whaling Commission that regulates all of this is constantly arguing about whether they should permit the Japanese to continue taking whales, and why a number of countries that would like to resume whaling are constantly being denied the opportunity to do so, because even in the face of international treaties, it appears that the wrong whales from the wrong populations are still being harvested, even though there is an international ban on taking those. And you can demonstrate this by using DNA profiling. So there are a number of different ways in which you can utilize DNA fingerprinting. So let's take another quick break here before we move on to using DNA technology in a number of other interesting circumstances. So modern forensic scientists only require a small trace of DNA from a crime scene, as little as 15 cells worth, as I've been pointing out, for forensic analysis. Why is this the case? You should be able to recall that, I hope. What are STRs and why are they useful for DNA profiling? Remember, STR stands for short tandem repeat. See you on the other side of the break for the answers.